My name's Sarah and I'm in the Cancer Genetics Lab in Biochemistry, and I'm a second year master's student, and my supervisor is Anita Dunedin, who Meg mentioned in her talk. So a bit about how I got into biochem. So I'm from Hamilton and I went to Hillcrest High School, and I liked science all throughout high school. I took all three sciences every year that I could, and then decided I wanted to come down here for uni. And in my undergrad, I did a Bachelor of Biomedical Sciences, so I took biochemistry, pharmacology and pathology. So I wasn't actually a biochemistry major at that stage. And then when I decided that I wanted to do postgrad, I really enjoyed the research in the biochemistry department, and I really liked the department itself. So now I'm in the department for most of my time in the lab doing my research. So a bit of an intro. So I also work on breast cancer, and this is really prevalent, with about 2,500 women each year in New Zealand being diagnosed. And not only is it really prevalent, but it's also really important as it's quite severe. So it's the #1 cause of cancer-related death in women worldwide. And like Meg said, it's split into different subtypes. So this is based on the characteristics of the cells within the tumour. And the subtype that I work on is estrogen receptor positive. So these are two slides of tissue taken from normal breast and estrogen receptor positive breast cancer. And so what you're looking at here are the cells you would see if you looked at the tissue down a microscope. And the little brown dots are estrogen receptors. So as it's pretty obvious, in normal breast tissue there is a normal amount of estrogen receptors, and in estrogen receptor positive breast cancer tissue there are way more of these receptors. And so this is important because the naturally produced hormone estrogen binds these receptors and signals the cell to grow. So when you have a normal number of receptors, you get a normal amount of growth to help the tissue stay healthy. However, when you have heaps and heaps of these receptors, as in the cancer tissue, the cells have uncontrolled growth, which is essentially the basis of cancer. So based on this knowledge, targeted therapies have been produced, known as hormone therapies. So you may have heard of tamoxifen, which is an example commonly used for this type of breast cancer. And this essentially binds the receptor so that the naturally produced hormone can't. And if the hormone can't bind, it can't tell the cell to grow, which essentially should stop the cancer. However, this is all well and good in theory, but resistance to this treatment is still quite high, and we don't really know why. So this is where my project comes in. So I'm looking at a specific region of the DNA within these cells. So in this region is the estrogen receptor gene and 3 upstream genes. And previous research has found that a mutation that occurs here is important in terms of it increases your susceptibility to getting this breast cancer, as well as increasing the likelihood that you will be resistant to the treatment. And so to try and figure out how this might be happening, I'm also using CRISPR, but slightly different to me. So I'm using it to try and edit the DNA of the cells. So I have estrogen receptor positive cells, and I want to give them the mutation that I want to look at, so that way I can compare how they might be different and therefore how maybe the mutation is affecting the cells. So how this works is the CRISPR is made up of the guide RNA and a Cas9 protein. So the guide RNA guides the Cas9 to the section of DNA that you want to edit, and once it's there, the Cas9 protein cuts the DNA at that specific point. And now this cut DNA, the cell doesn't like it, so it wants it to repair itself back together. And during this process, you add in your template DNA, which has your mutation of interest. And so hopefully when the cell repairs the DNA, it'll also insert the mutation that I want to look at. So therefore I can look at the cells that have my mutation and the cells that don't, and try and figure out maybe what's happening. And the theory that we have at the moment is that it's affecting the chromatin. So as most of you know, DNA is made up of 4 different bases, and this is twisted into a double helix. But this double helix needs to fit into your tiny, tiny cells that you can't even see. And so how this works is that it's compacted really, really tightly, so it's wrapped into little balls, and these little balls of DNA are then wrapped around themselves again. And this structure is different in every cell, depending on what bits of the DNA the cell needs to survive. So open chromatin means that that DNA can be used by the cell. So the theory is that my little mutation is changing the shape of this chromatin, which means that different bits of the DNA are available to the cell, and maybe this is how the cell is acting differently. But I'm still working on that, so yeah.